And now, it's my great pleasure to introduce Dr. Marina Caskey to you. She's currently an associate professor at Rockefeller in the Laboratory of Molecular Immunology. Marina hails from Brazil, where she graduated from medical school in 1998. She completed her residency at St. Luke's Roosevelt Hospital here in New York City, followed by a fellowship in infectious diseases at New York Presbyterian Weill Cornell Medical Center, where she still serves as an attending physician. Marina came to Rockefeller in 2003 to join our Clinical Scholars Program, a 3-year master's degree program that prepares physician scientists for careers in translational medicine. Marina studied with the late Nobel laureate Dr. Ralph Steinman. Her research focused on the characterization of the immune response induced by an experimental vaccine that targets HIV proteins directly to dendritic cells. After Ralph's death in 2011, Marina continued her research with Dr. Michel Nussenzweig. Marina and Michel have teamed up to use innovative basic science and expert clinical research approaches to create a novel antibody treatment for HIV infection. Marina will describe this work during her talk this afternoon. Marina's seminal studies have shaped this new area of antibody research. Several groups in the US and abroad are now studying the antiviral and immune-modifying effects of these molecules in combination with other therapeutic strategies. Marina is currently a part of a team heading a new COVID-19 clinical trial at the Rockefeller Research Hospital. The purpose of this trial is to identify antibodies from the plasma of COVID-19 survivors that have the ability to neutralize the virus and prevent it from infecting cells. These elite antibodies hold potential to become potent drugs to fight or prevent COVID-19. Before I hand over the podium to Marina, I once again would like to thank all of you for your participation this afternoon, and also for the questions that you sent in to us in advance of this discussion. During the program this afternoon, you're welcome to ask more questions. You can do this at any time by clicking on the Q&A icon that you can find at the bottom of your screen. A box will open and allow you to type in your questions there. We'll have time for questions and answers following Marina's presentation, and we will do our best to answer as many of these questions as we can before the end of the hour. And now it is my great pleasure to invite Dr. Marina Caskey to join us. Welcome, Marina.
Thank you, Rick, for the kind introduction and for the opportunity to speak to all of you today about my work on HIV and how we have, in the past 1 to 2 months, refocused our efforts to address the challenges of COVID-19. So what I thought I would do today is to start by discussing with you the potential roles of passive transfer of antibodies against infectious diseases. And for that, I'm going to use as an example the work that we have done over the last 5 to 6 years with neutralizing antibodies against HIV. And then I'll share what we've been able to accomplish in this past month with our efforts to identify potent antibodies against SARS-CoV-2, which is the coronavirus that causes COVID-19 and who is responsible for the present pandemic that we are all facing. So as many of you or all of you know, antibodies are these Y-shaped molecules, antibodies that fight infectious diseases. Antibodies are produced by B cells. So B cells are cells that are components of our immune system. When we face a new infection, these B cells respond to this threat by producing a whole array of different antibodies. And what the antibodies do, they are able to recognize specific portions of this new virus in a way that is very specific, meaning that the antibody, a portion of the antibody binds very specifically to very specific areas within the virus in a lock and key manner. And then by doing so, antibodies can then block infection. The same way that when we respond to a new infection, our bodies produce a multitude of different antibodies, the antibody that recognize different areas on a virus. Antibodies also possess very different functions. So there are antibodies that are, as illustrated here, that they can block free circulating virus from causing infection. So these are called neutralizing antibodies. And whereas there are other antibodies that what they do is they tag either circulating viruses or infected cells that express pieces of virus on their surface. And by doing so, it's a way for the antibodies to alert and engage the rest of the immune system, the rest of the immune cells, to come together and try to eliminate this foreign pathogen. The idea of using antibodies to prevent or treat infections is not something new. It's something that has been around since the late 1800s. And it's important to know that it's different. Passive immunization or transfer of plasma or transfer of antibodies is different than vaccination. When you vaccinate someone, you're trying to teach the immune system to produce antibodies, to induce antibodies to fight that infection that you mimic with a vaccine, so that your immune system can continue to produce those antibodies long term. In the absence of an effective vaccine, what has been shown time after time is that the transfer of pre-made antibodies can serve as a tool to prevent or treat infection. So illustrated here is the case of diphtheria, where in the early 1900s, when serum diphtheria antitoxin, which is a mixture of serum coming from horses, were transferred to infected individuals, the death rate of diphtheria in the US decreased tremendously, showing that how effective this kind of treatment was at the time. And that allowed sufficient time for a diphtheria vaccine to be developed, which further decreased the mortality rate to a point where we don't really think much about diphtheria these days. And diphtheria is just one among many infectious diseases that have been initially handled by transfer of antibodies before vaccines became available. However, antibodies don't come in different. They like I mentioned, antibodies come in with different functions. When you transfer plasma, you're transferring a mixture of antibodies that are not only antibodies against the particular disease of interest, and then even among the antibodies that are against that particular disease, there are antibodies that have different functions and activities. So it's known that a small percent of such antibodies have very high activity against a particular virus. And this is something that our lab and other groups have found when studying individuals that recovered or that were infected with viruses like HIV, Zika, and dengue, where it appears that exceptional antibodies can be generated, but in a small percent of these individuals. So in order to harness these antibody responses, the lab of Michel Nussenzweig developed a method that allowed the identification of these exceptional antibodies, which is a little bit like trying to find a needle in a haystack, because they are usually rare entities. So the way that this method works is that you start first by screening a large pool of individuals that have recovered from an infection or that carry an infection, a chronic infection like HIV. And you do that, and then you test the level of antibody responses that this large pool of individuals have against that particular virus, in this case, HIV. By doing that, you're able to identify those elite neutralizers, the 5% of individuals that have remarkable antibody responses. And then you go on from trying to identify the specific cells within their blood that are producing antibodies that are able to recognize a very particular part of the virus. So the way that this is done is the bulk of their B cells are mixed with proteins from the virus, from HIV. And that protein serves as a bait to which the B cells the B cells can recognize, bind to, and then be sorted out, be fished out of that pool of other B cells into a plate, one by one. And once that is accomplished, in the laboratory, the genes that produce the antibodies that those B cells are made can be sequenced, cloned, and these antibodies can then be produced in the laboratory. And that really allows the isolation of hundreds or thousands of antibodies. So you can generate a large library of antibodies against HIV in this case. And you can go on to characterize these antibodies in very great detail, with the idea of selecting the most potent ones to further characterize and to bring forward as candidates to testing in other laboratory models and in potentially in clinical studies. So through methods that like this that were developed here and adapted by other laboratories, many anti-HIV neutralizing antibodies have been discovered over the last 10 years. What this figure here is showing is the HIV envelope protein, which is the protein that the virus expresses on its surface. And similar to the spike protein of coronavirus that you might have heard about, it's this protein of the virus that makes contact with the human cell, giving entry of the virus into the cell to cause infection. And the colored areas within this protein are areas against which antibodies, neutralizing antibodies, can be produced that will then interfere with the ability of this virus to infect cells. So here at Rockefeller, Michel Nussenzweig and his colleagues identified two antibodies that are highly potent against HIV and bind against two specific areas within this HIV envelope protein. And then these antibodies are illustrated here. So this figure to the right shows the structure of 1074, the blue antibody, and 3BNC117, and how they interact with the HIV protein. So this comes to show that these two antibodies, even though they are HIV antibodies, they attach, they identify and bind to the virus from different angles. And what that results is that when they are given in combination, they are able to demonstrate activity to block the virus from causing infection in more than 90% of laboratory strains when they were tested. So these antibodies that are very potent and very broad seem to be exciting new candidates to be evaluated as potential new modes of treatment or prevention against HIV. So you might ask yourself why, what would be the utility at this point of develop new modalities to treat or prevent HIV? It is true that antiretroviral therapy is highly successful, and it prevents, and indeed it prevents the progression from HIV to AIDS. However, there are significant challenges in the HIV field that remain and that have been very difficult to address. Very significantly, the number of new infections per year has remained very high. In fact, it's plateaued over the last several years, where close to 2 million people are newly infected each year. And then unfortunately, we still do not have an effective vaccine to prevent HIV. And while ART or antiretroviral therapy is highly efficacious to delay or prevent disease progression, there is no cure for HIV. So once HIV infected, the person needs to take those medications for life. So with the overarching goal of evaluating if these antibodies indeed would show to be safe and also to be efficacious in interfering with HIV infection, we went on to launch a series of early clinical trials to test these antibodies that were discovered here in both HIV, in people living with HIV, as well as in HIV uninfected volunteers. And these studies were conducted at our hospital here, which is a hospital that was founded in 1910, and then it's a hospital where all the activities in the hospital are directed towards patient research. So this environment really provides the ideal situation for us to effectively, quickly conduct our clinical trials. Since 2014, so over the last 6 years now, we have conducted about 10 clinical trials using these two antibodies in different clinical scenarios. And what we have found is that when we gave either one or these two antibodies in combination to people living with HIV that were not on treatment, so they had circulating virus in their blood, the two antibodies showed very high activity against the virus, lowering the amount of virus circulating in their bloodstream significantly to levels that were similar to some of the antiretroviral drugs. In addition, when we looked at individuals who had been on antiretrovirus and then had achieved undetectable viral load, so they were successfully treated with antiretrovirus, and we transitioned them, meaning we stopped their antiretrovirus and instead gave them treatment with the combination of antibodies. We showed that these antibodies were successful in maintaining their virus levels suppressed, very low levels, similar to what antiretrovirus could do, as long as we maintained dosing the antibodies. And more recently, we have tested long-acting forms of these antibodies, and then we found that these antibodies can indeed maintain therapeutic levels for a very long time, which suggests that they could potentially be used as infrequently as every once or twice a year. Given these promising results that we showed with our studies here at Rockefeller and other colleagues elsewhere, antibodies now have really taken center stage in the HIV field, and they are now being tested in many different clinical situations. So they are being evaluated as potential tools to prevent sexual transmission or mother-to-child transmission of HIV. They're also being evaluated as potential alternatives to antiretrovirus in situations where compliance with daily medication can become a great challenge. And most excitingly, they are being evaluated as potential components of strategies that aim to achieve an HIV cure or long-term viral control. And it's believed that antibodies could have significant effects in this context, because antibodies, like I said, can modify the way that the host, the person, controls the virus. And by doing so, it may be that the antibodies could interfere with the amount of antibody that remains despite successful antiretrovirus. So it's been a very busy and exciting period of time for us in the lab. And I'm using this slide here to illustrate how long it typically takes to develop something from the lab to the clinic. So usually it's a long process. In this case, it took about 10 years from developing a method, discovering the antibodies, testing the antibodies first in laboratory assays and laboratory models, producing the antibodies in large quantities and in a way that they could be safe enough to be given to humans in clinical trials. We entered clinical trials, and in the last 4 years, we generated sufficient data to support their testing in larger studies. So in fact, these antibodies are now being considered to be tested in a large prevention study, where we aim to show if these antibodies can indeed prevent a new HIV infection in Africa. And earlier this year, the antibodies were licensed to a pharmaceutical company, to Gilead, with the plans that they will take the antibodies into further later development. And we hope that these later studies will continue to show that these antibodies are indeed efficacious, and then they will hopefully have a role in HIV therapy and prevention. So while our clinical group was working on the HIV story, others in Michel's lab were working with studying antibody responses to different viruses like Zika, hepatitis B, etc. And they have successfully, again, identified very interesting and potent antibodies against these viruses. And these antibodies are now at the different stages of this antibody development. And very recently, with the emergence of SARS-CoV-2, it was very clear that we now needed to refocus our efforts and leverage all of the resources and technology that had been put in place for HIV and for other viruses to try to understand immune responses against SARS-2, and then hopefully be able to identify exceptional antibodies against this virus as well. So coronaviruses are RNA viruses, just like HIV, but they are different from HIV in that HIV causes a chronic infection. So once you're infected with HIV, your immune system is not capable of eliminating completely the virus. Whereas coronaviruses cause more of an acute infection, meaning that an individual can recover and be cured from the virus. The coronavirus, like HIV, this is a structure illustration of the virus itself. Like HIV, it expresses proteins in its surface, and it's this protein called the spike protein that binds to the cell in a human being, a person, and this protein gives entry, allows the virus to invade the cell and cause infection. Coronaviruses is a large family of viruses, as is illustrated here in this tree. There are coronaviruses that are usually in circulation, and they're cause of common cold. But in the last 15 years, we have seen the emergence of other coronaviruses that are more pathogenic, meaning that they can cause more significant disease in human beings. And most recently was SARS-2, which is the virus that we are all dealing with for the last several months. And SARS-2 is very closely related to SARS-CoV-1. So, like I said, we once it became clear the threat that SARS-2 was to us and to the world, in a great team effort, the laboratory and our clinical group refocused to try to ask and see if we could indeed identify exceptional antibodies against SARS-2 as we had done for other viruses. So we adapted and implemented the same methods that had been in place already, where we started by screening samples from recovered individuals, and with the idea that we would be able to find and produce a large library of antibodies that could then be studied individually, with the idea that we could then select potent candidates, meaning candidates that showed very high activity against the virus, that could then be tested further in the laboratory and hopefully be produced in large enough quantities to be tested in clinical trials and eventually to be used as a tool against this virus in the clinic. So in order to do that, we launched a study here to recruit volunteers, like Rick mentioned, who had recovered from COVID-19. And in the laboratory, part of the project was really spearheaded by Davide Robiani, who is also within Michel Nussenzweig's laboratory. We were really very fortunate in our recruitment efforts, such that in only a few weeks, we reached our target enrollment goals. And what I tried to do here is to show a map of where the volunteers that reached to our recruitment office in our first week of recruitment, where they were coming from. And they are coming from really the greater New York City area, with a great representation of residents of New Rochelle, which, as you remember, was the area where the first cases in New York State were reported. And then we also had individuals that resided in Lower Manhattan, in Brooklyn, as well as in areas of New Jersey. And besides the willingness of volunteers, that was really striking how all of the volunteers that approached us, they really wanted to be helpful. They felt so fortunate to have recovered from the illness that they really wanted to give back and be helpful in any way possible. In parallel to that, we wouldn't have been able to so quickly recruit so many participants if this hadn't become a big team effort that involved both the recruitment and facilitation office, as well as our nursing staff, that despite all of the challenges that we all have been facing in these last several weeks, we all came together and we were able to bring all of these volunteers in. So in addition to the efforts in the hospital, there was also a huge effort from the laboratory side, and we are lucky that we work in very close connection with our bench colleagues in not only Michel's lab but also other labs on campus, like Paul Bianash's and Charles Rice's lab, and they all came together to quickly translate and work with the samples that we had collected in the hospital to analyze antibody responses, and then get to what I'm going to share with you today. So in our study, our goal was to we set out to enroll both individuals that had been formally diagnosed as having COVID infection with the PCR test, but also we thought it would be interesting to bring in individuals that had been exposed, they had clear exposures to COVID positive individuals, and they had also developed symptoms. So we call these contacts because they hadn't been formally diagnosed with COVID-19. But as it turns out, the two cohorts turned out to be very similar, which really represents that our contacts were indeed individuals that had COVID-19, but were unfortunately not diagnosed because of the restrictions in testing that we were facing at the time. The two groups were very similar in age and then gender distribution. The volunteers on average presented for sample collection approximately 4 weeks from onset of their symptoms. And on average, they experienced symptoms for about 10 days. And in this initial cohort, the participants, the volunteers that we recruited, they really experienced more of the mild to moderate versions of COVID-19. And they did not require hospitalization. And I included this graph here just again to illustrate how the symptom severity reported by what we called our cases and our contacts were very similar. So in reality, we pulled them all together as individuals that had experienced infection by SARS-2. And the symptoms that they experienced and then reported to us were the typical symptoms that have been reported elsewhere, where most common symptoms were fever, dry cough, body aches. And we also found that a relatively low percent of individuals had comorbidities in our cohort, which makes sense when we think that we were really, in this initial set of volunteers, we had recruited people that had experienced mild to moderate disease. And what have we found so far in terms of their antibody responses? So our colleagues in the laboratory set out to measure the antibody levels against specific areas within the coronavirus, specifically against the receptor binding domain. And what this graph here is showing and illustrates is how the levels of antibodies really varied in our cohort. The three black lines are negative control. So these are samples from healthy volunteers that were collected a few years ago. So they were negative for coronavirus. And the gray, very thin lines are the spread of our recovered COVID participants. So some of them had really very low antibody responses, but we did have some outliers that had higher antibody levels. And interestingly to us, we found that there is a strong correlation between the duration as well as the severity of the symptoms that they reported with the level of antibodies against COVID that we measured. But like I mentioned earlier, not all antibodies are the same. So besides measuring the amount of antibodies that they built against SARS-2, we're interested to know the functionality of the antibodies, specifically if within the samples of a mixture of antibodies, if these antibodies had indeed neutralization ability against the virus, if they could really block the virus from causing infection. So in order to do that, Theodora and Paul Bianash's group, they developed an assay that allowed us to test that in the laboratory in a plate. And the way this works is you mix either antibodies or plasma and with pseudoviruses. So pseudoviruses are lab strains of viruses that express specific portions of SARS-2. And then the reason that we do that in the lab is because it facilitates the handling of the samples in the lab over what would be required to do with the real coronavirus, so to speak. So the way that assay works is you use these mixtures and you incubate, you put them together with cells that can be infected by SARS-2. And these cells are modified in a way that when they are infected and they start to produce virus, they light up a color that then can be measured. So that allows us to see how much infection you can cause by your mixture of antibodies and pseudovirus. So the idea here is that if you have antibodies that are effective in blocking the virus from causing infection, you're going to have a very low signal of infection. And that allows you to also know how much of a given antibody you need to add to block infection. And that amount is what we refer to as a neutralization titer. So when we went, the samples from our first almost 70 participants were tested for this neutralization activity of their plasmas. And what we found was a little surprising, was that almost 20% of the samples tested did not have any detectable neutralization activity. So they had antibodies, but they didn't have this functionality of being able to neutralize. And among the ones that had detectable levels, there was really a great variability on the level of neutralization activity, with very few individuals having high levels of neutralization activity in their plasma. However, one thing that it was interesting to us is that here, the level of neutralization activity, so the activity to block the virus, correlated with the amount of antibody against the specific portion of SARS-2. So perhaps this can be an indicator. Levels of against the RBD region of SARS-2 can be an indicator or a correlate of neutralizing activity. But if you remember from what I described earlier, our goal really is to identify the individual antibodies that are responsible for this high neutralizing activity. And in order to do that, we went to the blood samples in order to isolate single B cells that could recognize in a very specific way this receptor binding domain, this specific area within the coronavirus. And then by doing so, we are able to produce hundreds of antibodies so far. And what these pie charts here are representing, each pie chart comes from a sample of a different volunteer. The number in the middle represents the number of SARS-2 antibodies that we are able to recover. And each slice of the pie with these colors represents families of antibodies that were recovered. So what this illustrates is that each individual can make a multitude to different families of antibodies, all recognizing SARS-2, but in different ways. And same families, like for example, the red clones or the red families of antibodies can happen in different individuals, which suggests that there are similar pathways leading to antibody responses that are triggered when a person is infected with this virus. So similarly to what we did with the plasma, with the whole mixture of their antibodies, we're interested to now understand across the hundreds of antibodies that we recovered, how active each of those antibodies were. And then again, similar to what we had found with plasma, we find that the different antibodies have very different levels of neutralization activity, with really only a few of them being highly active. But even though they are rare, we were able to find some of them that were indeed exceptional and highly potent against the virus in the nanogram range. So in a very minuscule amount of the antibody was able to block infection in that laboratory assay. So that's where we are. So what we know so far from the initial cohort that has been analyzed is that individuals that recovered from mild to moderate COVID-19 showed variable levels of neutralization activity in plasma. And despite the overall low plasma neutralizing titers that we found in our cohort, we were successful in identifying individual antibodies that showed exceptional activity against SARS-2. And where do we go from here? We have many more samples that are now being tested with the hope that we will be able to find other exceptional antibodies. And the antibodies that have already been found, we are trying to understand how these antibodies find and bind to the virus, because we are looking for antibodies that can be complementary to each other. Remember that for HIV, we showed that a combination of two antibodies was better than only a single antibody, and then it may be that's the case for coronavirus as well. And we are going to continue to test these antibodies to see if in other laboratory models, the antibodies can indeed prevent or clear infection. Usually, you do all the laboratory analysis before you embark into a clinical development of antibody candidates. But here we are really trying to shorten our timelines. And for that, we have decided to already begin the clinical development of some of these antibodies that look very promising to us. And the way that we are doing this is we are starting the very first steps to manufacture, to produce the antibodies in a way that they can be given to human beings. We have started to plan the first clinical trials, and we also initiated conversations with the FDA to see how we can bring these antibodies into the clinic. And with that, like I mentioned throughout my talk, this is really a large team effort led by Michel and Davide Robiani in the lab. It took really the entire lab and our processing lab and clinical team to put all of this together in just 4 weeks. With great collaboration from the BNASH and the RICE labs, as well as with colleagues from other institutions like Caltech and the Chan Zuckerberg Biohub. And in addition, we had tremendous support from different departments within our hospital. And I'd like to especially thank Arlene Hurley, who really spearheaded the initial recruitment efforts and of bringing these patients in, these volunteers in. And with that, I'd like to thank the support that we have received for this project and other projects in the lab, in particular the Robertson Development Fund, as well as Robert Wennett. We receive also support from the Gates Foundation and from NIH. And with that, I'd like to especially thank the Women in Science donors. It's disappointing, like Rick said, that we are not together in person, but I hope that I'll be able to see you next year for next year's luncheon. So again, thank you for your support and then for listening to my presentation today.
Thank you so much, Marina. That was a truly spectacular presentation and incredibly informative and impactful work. Congratulations. So we now have time for questions and answers. Many of you have written in questions already, but again, if you would like to ask new questions, please go to the Q&A button at the bottom of your screen and you can type in your questions there. And we'll get through as many of these as we can in the time that we have. Marina, maybe a first question for you would be, what do you see as the most important clinical uses for these neutralizing antibodies to SARS-CoV-2 virus?
I think, Rick, that I think that they could play a role both in prevention as well as in therapy, but I think that they could be a very practical, perhaps, tool to have in the prevention of infection of highly exposed or vulnerable subpopulations like healthcare professionals or residents in nursing homes until we have a vaccine. So I really think that it can be an important bridge from now until we have an efficacious vaccine.
So imagine that we have a vaccine come out. Do you think these antibodies may still play a useful role? I can imagine, for example, in the elderly population that doesn't mount as robust an immune response as frequently, that maybe these would still have an important role, for example, in nursing homes, as you suggested.
Yes, I agree. That would be one scenario. And I also think that another scenario is for what's called post-exposure prophylaxis. So someone who has not been vaccinated and has an exposure. If you are to vaccinate, it takes time for the immune system to build that immune response. Whereas if you transfer antibodies, you have the tools, you have the immune response there available to prevent the infection.
Great. You mentioned that in the cohort that you've studied already, that the longer the symptoms persisted, the higher the antibody response that you saw. So I wonder if when you look, you haven't looked at people who were hospitalized and had more severe manifestations of their disease. Do you have speculation as to whether they will have higher antibodies because they've had longer exposure, or fewer antibodies and that's why they got sick, more sick?
So that's a good question. We now have a cohort of individuals that required hospitalization. Very early analysis suggests that they have significant antibody responses. What we don't know is so these were individuals that were required hospitalization, required treatment, but they recovered, and they didn't become critically ill. They didn't become the individuals that really required long-term intubation, etc. I'm not sure. So I think that more severe disease, we will see that is linked to higher and perhaps better antibody responses, but to a certain point. And maybe that the super sick, critically ill will not be able to mount these responses, but we don't know. When we decided to also look at exposed individuals, we thought, well, perhaps people that are exposed but that don't develop the infection, or that don't develop a lot of symptoms, are the ones that will have built very interesting antibody responses. But that's not what we found. And to me, that probably suggests that it's not only about antibodies, there's also the T cell component of our immune system that is playing a role in how we clear the infection.
Great. So a number of people asked about COVID-19 in children, with the observation that at least initially it was observed that children almost never get seriously ill. And now we recognize that there's a late inflammatory disorder that's appearing rarely in these children. What do you think explains the difference in how children and adults respond to the virus?
I don't know if that's well understood yet, or I don't understand it well yet. Children have a more flexible, so to speak, or a less mature immune system, and perhaps they're able to more easily adapt to and respond to viruses that they're exposed to. Perhaps that's what happens, and that's why they experience less severe disease. What we are seeing now, this post-viral inflammatory serious illness, I don't know if it's antibody driven or what's really driving this exaggerated inflammatory response that a rare number of children are experiencing. So in short, I'm not sure.
So thank you. There are a number of questions as well about the immunity, and your presentation highlighted this, that among people who have recovered from the virus, many of them appear to be making no neutralizing antibodies at all. So now that we're starting to get antibody tests that we can all go out and see if we've been exposed to the virus, what do you think that tells us about our future immunity to infection?
So I think that, but in our cohort, right, and others have demonstrated similar things where we know that the individual recovered without treatment. So the immune system did build a immune response that was able to clear that infection, which may not have been solely antibody mediated. So I think that's how we explain that part of it. Now, what do the antibody tests then show? To me, what we are finding and what the antibody tests show is that I believe that there is at least a certain level of protection when you do build antibody responses, but it may be that this protection is not complete. It may be a partial immune response, and then we don't yet know how durable that immune response is. So, so I think that we have to continue to be very vigilant and not think that a positive antibody response equals complete immunity.
Do we have insight from prior experience with other coronaviruses that might be helpful in understanding how long immunity might last?
So with other coronaviruses, it seems that antibody responses can last for a few years, and then they wane. And you can, and for the common cold viruses, you can acquire those viruses after these antibodies went to very low levels. With this new coronavirus, it's all very new, so we don't fully understand yet.
Does anyone have an understanding of why so many of these coronaviruses exist in bats, and why do bats apparently not become ill from these viruses?
I don't know if I know the answer to why the bats don't become ill from the viruses. Perhaps you know that. Um, I think
Why
that
do you think I asked you?
I don't know. I think that we are seeing, I've read that perhaps we're seeing more of these transitions from viruses that circulate in bats and animals because, not only the world population has grown, but we live perhaps in certain areas of the world in closer contact. So the environment, the has changed where the habitats of human beings and these animals are coming closer together, allowing more contact, and then risking these changes in transitions, transfers to happen.
So this is the third coronavirus that has jumped from animals to humans in the last 20 years, and raises the question of whether we're just going to have a constant stream of these finding their way into the human population. That we're able to unify against coronavirus, or them make antibodies to be broadly neutralizing any of these.
So what we were hoping to see with the antibodies that we have identified was to, remember how the virus, the three viruses that emerged in the last 15 years, they are related to each other. They are not identical, but they are related to each other. So we are hoping, and we still are hoping, that we will be able to find antibodies that can have activity across these different highly pathogenic antibodies. The ones that we have found so far, they don't seem to be that way. They seem to be very specific against SARS-2. So I think that if it's expected, or there is a real concern that this will happen again with new viruses. And I think that what we need to learn from this experience now is to be prepared and adapt. The research world has really focused and generated a lot of knowledge about this new virus in just a few months. And I think that the resources that are put in place now, they have to be maintained in such a way that we can quickly adapt and then respond to a new emerging virus that might arise. If a vaccine will be a once in a lifetime vaccine, or if it's going to have to look more like an influenza vaccine where it needs to be tweaked every year, I think that still remains to be seen. This virus seems to not mutate as rapidly as influenza, so perhaps we are not going to be looking at an yearly new vaccine.
Great. Are there side effects from these antibodies that you might be concerned about when you go into phase 1 clinical trials?
That is a very good question. So we have given the monoclonal antibodies in HIV to hundreds of people now. And in general, they have been very safe and very tolerated. However, with SARS-2, there is a concern that with this particular virus, antibodies that are highly neutralizing. It's expected that these antibodies are going to be safe and then they'll clear the infection. However, there have been laboratory studies suggesting that if you have less than ideal antibody response against SARS-1, that you could actually worsen the progression of the disease. We don't know if that's going to be the case with SARS-2. So typically, monoclonal antibodies are very well, they're safe and very well tolerated. With this particular virus, because of what's known with the earlier species, the studies have to be done very carefully as to avoid what's called antibody-dependent enhancement, if the antibodies are not at a high enough dose or potent enough to completely block the antibody. So that's one challenge that we have to deal with, the same way that the vaccine trials will have to be careful about this potential challenge.
Is it clear that, you know, one of the clinical hallmarks of COVID-19 has been the relatively late acceleration in the severity of the respiratory disease? And there's been a lot of speculation that this might relate to the development of the immune response to the virus. Is there evidence one way or another at this point that you find persuasive as to whether it's the immune response that's causing the worsening respiratory symptoms?
I don't think that there is any direct data proving that that's what's happening, but it seems to mimic what's seen in some cancer treatments with T cells, that what's these called CAR T cells that can induce this very exaggerated inflammatory response that leads to what's called the cytokine storm that causes the individual to be very sick. I don't know in the context of COVID-19 if it's already known for sure how the pathogenesis of progression actually happens.
You've mentioned that you've already studied over 150 patients and their antibodies. Have you noticed any difference between men and women, for example? There's this striking difference in the mortality difference between men and women with who are infected with the virus.
No, their antibody responses were very similar. You know, during our interviews with the participants, it did seem in general that the men had experienced more severe illness than the women, even when they were, household, husband and wife live in the same house. So presumably having been exposed to exactly the same virus. But that was not reflected in their antibody responses. The antibody responses are very similar between men and women.
In the case of HIV, it's been known that there are some patients who can't be effectively infected with the virus at all. Do you have any evidence or suspicion as to whether there might be patients who are completely immune to infection with the SARS-CoV-2 virus?
So again, maybe I think that Jean-Laurent Casanova is studying these outliers, both individuals that did not have were young and did not have comorbidities and shouldn't have experienced very severe disease and yet had fulminant cases of COVID-19. And I believe that now he's also going to be looking at the individuals that did not develop infection. So I think there are reports of household contacts, healthcare workers that despite multiple exposures have not been infected or diagnosed with infection or developed disease. So there may be such a thing as being protected from infection, but we just don't know if that is true. And then if so, what is the mechanism?
Antibodies are a qualitatively different modality than most therapeutics, which are small molecules. Can you combine antibodies and small molecule therapies?
Yes, I think that's what we have been doing and thinking about for HIV, and I think the same can be applied to this infection. But I think that one thing that is important to note, like you said, is it's the early phase of the disease is driven by the virus. And then it seems that at one point, this exaggerated disruptive immune response takes over, and then that leads to the very severe critical illness. So I think that both in the context of the antivirals against the small molecules that have antiviral activity against the virus, and in the case of antibodies, the ideal situation for treatment would be to use these modalities earlier in the course of the disease, since you really want to intervene with the virus, at the virus, to prevent then the exaggerated immune response that happens later.
So that gets to an important question about testing and whether we have enough tests to be able to identify people who are early in the course of their infection. Up to now, we've been mostly identifying people when they get sick enough to need to come to the hospital. Do you have thoughts about how we should be, are we doing enough testing? And if not, how should we be trying to use the capacity that we have?
I think we are not doing, clearly we are not doing enough testing. I think this is going to be particularly important when we think about reopening. I think it will be important to, as we reopen, to do contact tracing, where you really try, once someone is identified with an infection, you really need to try and test their close contacts. It would be helpful if new testing modalities were to be developed that are faster, easier to run type assays. And again, like in every aspect of the virus, I think there are many research groups around the world that are working on this, including here, trying to develop new testing modalities that are simpler, that can be point of care, meaning that can be a rapid test, and then hopefully can be implemented in higher numbers.
Wonderful. Well, I'm sorry to say that we are at our appointed time to end our discussion. I want once again to thank Marina for a wonderful overview of her work. And as you've heard, she's made remarkable strides in just a very short period of time. I also want to thank all of you who have participated by attending today, have asked such insightful questions, and who have provided such generous support to the Women in Science initiative. We still have a lot of work ahead of us, and your philanthropic support continues to be essential to our progress. Please see the next slide for more information about how you can contribute to the Rockefeller University to help combat the COVID-19 pandemic. Thank you once again for joining us this afternoon. I hope you will continue to join us for future virtual discussions. Until then, all of us at the Rockefeller University hope that you will stay safe and be healthy. Good evening.